Hey, I'm Fabian from the Ehrenreich Lab at USC, and today I'm going to be telling you a little bit about my work in sequencing the spadefoot codes via bioinfo and Seidl. We're doing this work in collaboration with the Fabian labs at UNC, who really pioneered all of the work in this organism, and so a lot of thanks to them. So in my lab, what we really care about is trying to dissect complex genetic traits down to a gene level, and we've had particular success with traits that can be at least partially binarized. And here what I'm showing is two spadefoot tadpoles from the same cross performed in lab by our collaborators. The larger one, the difference being that the larger one was fed fairy shrimp early in development, and the smaller one was raised off plant detritus. And so there's this phenotypic switch in these organisms, which ends up producing carnivores, and it's specific to the Spia genus. We didn't see it, or it can't be observed in the Scapiopus, the most closely related genus. And particularly, two of these species seem to be ideal for studying this trait. The reason being that they occur in a very large range across the western United States, all the way down into Mexico, and they differ in their fraction of carnivores. And what's fascinating is that when they're in sympatry, there seems to be a switch where the Opicata reduces and is kind of pushed into the omnivore niche, and the Omicrons is forced into the carnivore niche. So these organisms can be crossed in lab and can be kept in lab, but the genomic resources aren't fully fledged in. So to address this, we prepared Nextera shotgun libraries, 100 by 100 base pair insert, 150 by 150 base pair insert, and synthetic long read libraries by aluminum. We assembled the Nextera and soaked the novo and the synthetic long reads in the provided assembler on base space. And then we input all the contigs from that into Genius assembler. That actually improved our contigs pretty massively. We went from a largest contig of about 10 kb to 40 kb, but it's still not ideal. It's still fairly fractured. And when we took a look at the camera profiles of the reads, we found that there was this repeat, which was present in nearly 80% of the reads of the bombifrons SOR like that, sorry, 70% of the bombifrons, and 80% of the multiplicata. And we think that that might have something to do with how fractured the genome is. So to address that, we're currently prepping nanopore and PacBio that will hopefully let us finish the genome. But in the meantime, because we had decent contigs, we decided to check out the gene content. We blasted the X tropicalis mRNAs versus our contigs, and we found that roughly most of our mRNAs were only about 40% matched. But what gives me confidence is on manual inspection, it seemed that there were multiple exons from the same mRNA on a single contig, and the patterns roughly match between the two species. So that makes me think that this isn't just noise, it's actually biological. We proceeded to kind of check out heterozygosity because it seemed to be a problem in our assembly as well. And we found about 0.7 sites per kb were heterozygous in bombifrons and 0.5 in multi. And what's also kind of nice is that there's a little bit of a drop in heterozygosity in all the contigs that we could associate to genes via the blast. And so we took a filtered set of the genes we were most confident in, did a DNDS, and found a small portion of genes that seem to be under positive selection. And when we compared the superset sort of these genes from comparisons between our Spia and Tropicalis and our two different Spia, it seemed that there was a set of 93 genes that occurred in both sets. These might be interesting targets to start chasing in terms of finding interesting biology. So with that, I'd like to thank you for listening. Thanks to my lab groups, and thanks to funding from NIH.